In this video, we're gonna focus much on genetics. Some students find it difficult to answer or even study the topic on their own. When I find a number, couldn't over something like that. So I will try my best. You should know some of the basics which are in genetics. So let's start. Some of the genetics and adule pakati apa, and we should only focusing on the left side here. Palimauti gene mwina munamvapo olo ini nomwe munayankhulapo kuti munthu uyu watenga majinzi a abambo wake, mwana uyu watenga majinzi a amayi ake, kaya ndi ulemu, kaya ndi mwano, kaya ndi mayendedwe, or even beauty. Mukutanthauza yani pamene kuko chimene mukutanthauza apa mwachidziwikire. If I can do this illustration, so let's have my tendi my apa amene ndikuti it's a parent, and tendi mwanayo amene ndikuti watengerayo. So this one, that's our offspring. Ndiye mukuti mwanayi watenga mwano wa amayi yake, kapena nzeru kukongola watenga amayi yake. What it means is that this parent has transferred the traits from body mwakemu ndokampatsa mwanayi. Ndikuti apanga donated, this one has received those traits. So that process of transferring information, kapena kuti ma characteristics from parent to offspring, imene machidwa kuti heredity. So let's say kuti azakufunsa pamayi, so what is heredity? This one is defined as the transfer of traits or characteristics from parents to offspring. Mwina munaonapo structure of a chromosome kuti chimaoneka china chake something like this, and inside that ndimene timakhala ndi DNA molecule, and izizi zingane kuti zikuyimirira majani ma genes. So tingane kuti mwinaka ka gene for skin color, for height, for kayankhulidwe, beauty, skin color, something like that. So these things zitha kuchoka mwa parent, like I said, ndikupita kwa mwana in the process of heredity. Information imene yoyo kaya ndi nzeru zimene zapita mwa mwanazo kuchoka kwa makolozo zimakhala in these structures called genes. You see? So tingane kuti genes, tingane kuti they are messengers, they carry information about an individual. So we can define here genes as structures or hereditary messengers. Hereditary messengers that carry information about an organism. Yeah, hereditary messengers that carry information about an organism. Info yo kandi ku skin color, kandi height, imakhala mene tuli majinzimo. So that's the thing. Therefore titokubwera natsopano kuma watu a genetics. So our genetics is defined as how these traits or characters. So here should be characters. How traits or characters are transferred from parent to offspring. So this one is a scientific study. It's the scientific study of how traits or characters are transferred from parents to offspring. Zimatheka bwanji kuti infoyo ichoke umu ipite kwa uyu. So according to my dictionary, pomwe papa mauti genes wa definition yake amanena kuti these are basic unit of heredity. So basic unit of heredity as another definition of genes. So we can relate the term genetics and heredity. But I think above genetics is the study of heredity. Then kulu amene anayambitsa izi kuti moyo dzivuta zokuza ndi genetics and munyadira mati father of genetics akulu amene ndi Gregory. It's a Gregory Mendel, father of genetics. Ndamene anayambitsa izi anayandika dimba kake kumangosewera ndi maluwa kawo, so yochita ofuna kuti vutitsira moyo wapo. So that's the thing. Now my genes in biology, they are represented by letters. Ndi ma letters atokhala ma capital letters kapena ma small letter. Tingadziwe bwanji kuti gene is being represented by a capital letter and this one is a small letter. Tibwere pomwe paja masteni apo ndika mwana ndi lese kuti kutalika kwa mwanako is the same as ndi mmene mayi wa ankakulira. Tingane kuti nuna height ya mwanayi anatenga amayi yake. So lese kuti tindi gene T. Should I use T or H? I think I should take H as for height. And this one akuyimirira tingona kuti tallness. And in the small h, this one immediately shortness. So we have shortness there. Yeah, something like that. So what has happened here? Sikuti mwanayi anangochoka mwa mai wa direct ndi kutuluka huko. No. Komano panalinso abambo. There was a father who contributed for the production of this guy. Ndengati mukumbukira muno bwino mwina munaonapo ndipo munamva timanena kuti a human being has 46 chromosomes. Ukunso ndikuti kuliso 46. So meiosis imachitika ndikupanga have this number kuti khale 23, ukunso timakhala ndi 23. The same applies here. We have 23. Also we're gonna have 23. So after fertilization, fertilization ikachitika pamene tikhale ndi kamwanaka. You see? So let's say kuti this gene ya tallness inaphatikiza ndi gene ya shortness ya abambo, mwana uja ndokoneka wantali. Ready? In that scenario tingane kuti gene ya height yochokera kwa masteni is dominant over gene ya abambo. Chifukwa abambo sanaonetse ma effects awo mwa mwana in terms of kutalika. In other words, magene amaenda awiri. Tinganene choncho kuti machinza amaenda awiri. Ine ochokera kwa adadi, ine ochokera kwa mama ndikuphatikiza. And the combination hiyo ndi imene ingapange determine kuti offspring aoneka bwanji. So ngatanga oneke ngati uku kuposa tingane kuti this gene from mother is dominant. And imene sakuonekayo, imene sakupanga express in presence of this dominant gene, imene imachidwa kuti it's a recessive gene. Therefore tingane kuti we have dominant gene as well as a recessive gene. A recessive gene. What is a dominant gene? A dominant gene is a gene which masks, imapanga maski another gene. It's like kumpanga control kuti asaoneke. Whereas the recessive gene is the one which is masked, is the one which is controlled. Kabina, it does not express itself in the presence of the other gene. So dominant genes in biology are presented by block letters. That means napangira munja capital H. Whereas the recessive gene here, they are represented by small letters. Bas awonde kuti tafotokoza mbali yanga yokhudzana ndi dominant gene komanso recessive gene. And pomwe papa these genes titokhala ndi ma combinations osiyanasiyana. Titokhala ndi this combination capital H and capital H. Titokhala ndi combination hii capital H small h, and you can also have this condition here ma small h okaoka. Pale condition komanso mazygote which are formed through these conditions here. Magazaona kuti ma dominant gene aphatikizana ndikupanga form a character. Condition imene imachedwa kuti homozygous condition. Homozygous condition, and a zygote which is formed through this pair of gene oti ndofanana imene imachedwa kuti homozygote. So homo means the same, right? So you can see a pair of gene controlling a character, a pair of similar gene controlling a character. So we have a homozygous condition. Similarly here, this one is also a homozygous condition, because this one is a combination of recessive genes. So this one is homozygous recessive. Pomwe each the homozygous dominant because it's a combination of two dominant genes which are similar, also have a combination of two recessive genes which are also similar. Whereas this guy here, akulawawa condition hii imachedwa kuti heterozygous condition. It's a condition in which a pair of dissimilar genes, ma genes oti ndosiyana, akupanga control a character. So hetero in biology it means different. So homo same. So nakamba zoti ma genes amaenda awiri as you can see kuti apange control a character. There is a term which is used to define a pair of genes which controls a character. So that term which we use in biology imachita kuti an allele. So an allele tingane kuti a pair of genes which controls a character. Yeah. So zimana kuti ma genes amaenda awiri chifukwa timakhala kuti tikutenga ina uku komanso ina the other side from two different parents kuti akapange control a character. Imene ndimwe machidwa kuti an allele. So let me also introduce another term as per these things. Tekambeza phenotype komanso genotype. Kungoona ma genes azipanga combine kuti apange control a character. Imene imachidwa kuti genotype. You can see up geno regular mode gene. Genotype, this one tingane kuti it's a genetic makeup. Kabina kuti combination of genes. Combination of genes for a character. Ma genes akaziphatikiza ndikupanga character. Imene ndimwe tikuyipanga lifa kuti ndi genotype. Whereas the phenotype, we define this one as the physical outlook the physical outlook or final appearance according to Oyimbo, and this one is the fine appearance. Fine appearance due to combination of genes. Due to combination of genes. So let's have these biases. Okay. Nazaga mbazuri tindima genes awiri. Capital H ndi small h. Then nagamba zoti apa titokani ma combinations angapo ngalimene ndapangira makosana atatu alapa. So ndingane kuti through these two here we have 3 genotypes. Tindi type 1, type 2 and type 3. These we call them genotype, like a combination of genes. Whereas genetically tinene apapa the final appearance kapenda the physical outlook imene ibwere pafuta tapanga combine those genes imene ndime timaipanga alifa kuti that's our phenotype. For instance, timakamba zoti capital H akuyimirira tallness and small h akuyimirira shortness, and this gene here is dominant over this one. Ndekuti zygote alapa amene adzakhala pure tall, adzakhala wantali chifukwa ndi combination of yani ma dominant genes okhaokha. Whereas this one here adzakhalanso wantali koma osati pure chifukwa ali ndi recessive gene pambali apa. Komano akhala wantali chifukwa yani chifukwa adawawa ali dominant over this small h. The h yosadzaonekera. This one as well will be tall. So we have here tall and we have here tall. Whereas this guy here, the majority genes okha we're gonna have that one. So this is our phenotypes here. So phenotype imabwera after genotype. So makamba za pure mukazangoona magenes oti aphatikiza nofanana imene imachidwa kuti a pure breed. Nekuti apa we have a pure breed number 1 and another pure breed number 2. Ndi mwina mukhoza kumazimva ndikamafotokoza kutsogoloko komano that's the thing. To understand how characters are transferred from parents to offspring, in biology we use genetic diagrams, and we have 2 of them. We can either use crosses and sometimes we can use punnet square. So ndiambe ndikupanga cross diagram to show how heredity occurs. So ndigwiritsa ntchito magenes omwe ndinali nawo aja, capital H for tallness and small h for shortness. Ndejo mwende kufuna tekai ndi pure breed uyu ndi pure breed tipange cross ngati nkulu mena nkapangira a Gregory. So let's have these. We have capital H's tizipange cross ndi small h's. So let me have these. Tiapange cross awo. So what will happen here, our and my parents anga, we have parents there, and the meiosis will take place. So meiosis will lead to formation of gametes. So we have something like that. So we label there meiosis. Kubanga segregation, kubanga separate, kabina good formation of gametes. So these are our gametes. The same applies here. We're gonna have small h, and also we're gonna have small h. Now, what will happen next? It's fertilization. So, nkulu uyu abangana fertilize ndi ada awa. Ndekuti ajipatsa capital H ndi small h. Whereas the same guy here abangana fertilize ndi nkulu alabai amene akatipatsa capital H this small h. Ndikuti we are done with the fertilization of these. What about the adawa? Uyu ndikuti atopangana fertilize ndi nkulala vai thinking ndizambireso ifunika zikhale mu line uno bwino osali china chikapezeka uko. Yeah, ndikuti apa we gonna have capital H small h, whereas the same guy ndi nkulu uyu atipatsa capital H small h. Ndikuti kuchitika apa we label that one fertilization, zimakhala bwino mapanga label. So we have fertilization and these are our offspring. That's our offspring. So apa chimachitika apa it's like many mapangira expand kumasamu mwapatsidwa ma bracket awiri x plus y mukati musendi m plus n. So it's like x akati musana ndi mn. So I'm referring to this one as x kuti musana ndi mn. You see? So you can see zimeza chitika mini hope. Komanso next mudzatenga nkulu uyu akati musenso the same thing zomwe zilapozo kuti zikakupatseni these products. That's what happens. Now, since we were crossing my pure breeds, offsprings which are produced after crossing pure breeds, my offsprings amene wamachidwa kuti first filial generation. First filial generation. Nkunena kwina filial means offspring, like first offsprings. And amati F1, those are F1. So let's find the genotype of this. Combination ilipo ina, yes, it's the same as here. Is it the same as that? Is it also same as that one? So ngana kuti genotype ratio, genotype ratio, that's 1:1. Izonse ndizofanana. That's 1:1. What about phenotype ratio? Let's have phenotype ratio here. Phenotype ratio. Since capital H is dominant over small h, they would what will happen here? This one will be tall. Like I said, that was dominant over this guy. The same applies here. It's tall, and this one as well will be tall, and this final guy here also be tall. And they would in terms of phenotype, it counts 1:1. All into fanana. So mutafunsi dava maiso, what are the characteristics of first filial generation? They would gula mini wa tiana diminity first generation after crossing the pure genotype ratio imaka 1:1, phenotype isolation imaka 1:1, all are the same. In other words, they are all heterozygous. They are all heterozygote. That's the thing. What if we cross these offsprings? This one crosses is tengatulutse yani. So let's have here capital H small h. We have there capital H small h. So let's cross these. This one cross, and this time I'm referring these as my parents. Then meiosis ikachitike hapa, tupachitika meiosis, and we're gonna have capital H and abo tizakandi small h. Yachitika hapa label that one kuti ndi meiosis. Monetse kuti mukuzitsata, and meiosis lead to formation of gametes. So you can write there gametes. The same applies with the other genotype. We're gonna have here capital H, here small h, something like that. So next it's fertilization. So fertilization terenga bigewa aphatikizana ndi uyu komanso nkulu yemweyi akaphatikizana ndi uyu. you see. So apa combination omwe ibwera apa ikhala ma bigger h's, ma capital h okhaokha, whereas iyi idzalipatsa h1 nkulu ndi h1 ng'ono. What about if you take this guy, nkulu uyu ndi nkulu uyu, komanso the same guy ndi that guy, ndikuti apo we are going to have capital H. I think let's have these line. So you have capital H, small h, and these ones the small h's. Ndikuti apa yachitika ndi fertilization. We have fertilization there, and here we have our offsprings. That's our offsprings. So let's find the phenotype and the genotype ratios. So phenotype and the genotype. We have genotype here, genotype ratio. Genotype ratio. Combination ilikonso ina ukuyama capital H palibe ilipo imodzi, so we put it there 1. Tana nayo nanga apa combination imene iliponso ina you can see kuti kufanana ndi, so we can put it there 1 2 2 zilipo ziwirizi. What about this guy here? Genotype yomwe ilapa iliponso ina ilipo yokhala, so we can put it there 1 2 1. You see. What about phenotype? So here let's have phenotype here, phenotype ratio. In phenotype ratio tuone kuti the physical outlook ikhala yani. So we have here our combination of pure genes to, ndikuti this one will be pure to, this guy will be to as well, for the dominant gene H over small h. The same applies here to, where this guy here will be short. So physical outlook phe osazosokoneza tikuti phenotype, the physical appearance, phenophy zose zikamba ndi phi, something like that. Ndi mungodzo kuti we are talking about appearance. So phenotype ratio, if you can count ndi to 1 to 2 to 3, ndikuti we have 3, then we have 1 short there as 1. That's the thing. And you can see kuti tintuti offspring alabawa abwera after we cross the F1. So offsprings which are produced after crossing the F1, I mean we refer them as second filial generation, and wanyadira manguti F2 where filial akuimirira offspring. Ndi mutadzafunsidwa pamayeso without drawing a genetic diagram, amadzafunsa what are the characteristics of F2 generation, mudzangodziwa kuti they have a phenotype ratio of filial to 1 and a genotype ratio of 1 to 1. That's what happens here. So that's the thing about these things. What if you can have a punnet square? Punnet square is so simple. Chimene chimachitika ndigwetsa ntchito zomwezi muone kuti punnet square muoneka bwanji. So let's have this, something like that. Ndi timakhala ndi ma line apo. Yeah, so when nditakujambula china chake chioneka chonchi. So titenga pachele matenga ma heterozygous HH tukumpanga cross ndi HH. So we have there HH. Okay, so HH ndikuti we have that parent there and another HH this hapa. So after meiosis this side ndikhala ndi capital H small h, whereas this side after meiosis I'm going to have a capital H small h. So tentuone zimene zopanga na overlap. Tikaima mbox you can see kuti tikhala ndi akulawa. Ready? Then we're gonna have maichi okaoka akuluakulu. What about this box here? You see kuti tindi nkuluyu ndi nkuluyu. You see, capital H, small h. So we're gonna have here capital H, small h. What about in this box here? Nekuti tikandi bigger the bigger level. Nekuti we are going to have capital H, small h. And in this box here, nekuti tikandi akulawa ndi akulu amene ali apawa. Nekuti apo we're gonna have the small h's. So you can see kuti timakamba za genotype ratio as 1:1. So iti tilipo tiwiri pali nkuluyu ndi nkuluyu. And enawa alipo okha awo. That's the thing. Ndikuti umu mankhala mopanda kanthu omwe chimakhala chopanda kanthu ichi. So you can indicate sometimes timapanga indicate, and it's good kupanga indicate. Simathandize. So uzapanga indicate kuti these are your gametes. Gametes, and these are parents. That's how we do a punnet square. It's the same thing. So awonde kuti niyo zanu zayo patakhala kuti pamaiso ngakhala kuti zachitika penepo. My guys, I think ndilekere pompa the first part of the video. Next video ndikambe zokhudzana ndi ma variations, kuti what are the variations. We should meet in the next video. Commando, please also wala upanga subscribe to my YouTube channel, Unique Online Classes Malawi. And if you have any question in any subject, number langa that's 0882435562. You can join my WhatsApp platform via this number, and you will have a question in any subject. You will tandizana omeneko. Ine mundi tandiza, ine sonde kutandizana. I think you know what I mean there. So that's it for this video. Thanks for watching, guys. Let's meet in the next video.